So this is actually going to be a two-part lesson. We'll only get maybe done the first part of it today, and that'll be what all the parts of the cell do. And then tomorrow we'll go over how the cell transports materials, and then look at our pre-lab stuff for the cell anatomy lab, which we'll be doing on Wednesday. Okay, so we'll be back in the lab looking at the parts of the cell. Okay, this time in detail, looking at the parts. Okay, not like what we did at the microscopy lab, which reminder is due tomorrow. Okay, make sure you have your microscopy lab reports ready to go for the start of class tomorrow. Okay, so key points for today. We need to look at and understand the significance of compartmentalization in cells. This is an important word. Okay, compartmentalization means that in a eukaryotic cell, like we have, okay, because we're eukaryotic organisms, there are separate compartments or organelles within the cell, each one doing a specific job. Okay? Compartmentalization is a big deal because most of those jobs don't go well together. Okay? I wouldn't want to digest my food in the same environment where I kept my genetic information. Okay? Because I would probably digest my genetic information, and that would be unadvisable. Okay? So I wouldn't want to do that. So having separate compartments for each job is important. It allows each job to function at maximum efficiency, which makes eukaryotic organisms more efficient than prokaryotic organisms that do not have compartmentalization. Okay? So examples of prokaryotic organisms would be bacteria. So bacteria, very small, very simple. They don't have organelles. All of their cellular processes just take place in the cytoplasm. That's it. And all in the same place. So none of them work very well. Okay? All right. Know the role of each organelle, so we'll be going over that. Understand that cells are a complex system of interdependent, not independent, interdependent processes. That means they depend on each other. Okay? And not necessarily in a like 1, 2, 3, 4, 5 kind of way where process 2 only depends on process 1, okay, and process 3 only depends on process 2. Okay, they're interconnected. Right? Every process depends on several other processes in order to work. Right? And then, like I said, probably not today, but tomorrow we'll look at how materials are transported in the cell, across the cell membrane, and things like that. All right, so compartmentalization. Like I said, this is an important concept here. Cells are divided into compartments by membranes. So there's the cell membrane that separates the cell from everything else. But within the cell are a bunch of other membranes, each making its own organelle. Okay, so the nuclear membrane encloses the nucleus. There's a set of conditions that are ideal for maintaining genetic information within there. Okay? The lysosome has a special membrane that encloses the digestive enzymes because we wouldn't want those to get out. Okay? The mitochondria has a double membrane to help insulate the rest of the cell against the process that goes on inside the mitochondria. That's where sugars are burned, essentially. It's not obviously there's not a fire inside your cell, but it's where they're broken down, okay, and the energy is released. That produces a lot of heat. Okay? We need to insulate the rest of the cell against that process. I wouldn't want that process to be going on, again, where I might store my genetic information. I wouldn't want it to get overheated. So it's in a separate environment, right? So that compartmentalization that's present only in eukaryotic cells, okay, that's cells like we have, cells with a nucleus, provide different local environments that allow the cell to perform metabolic functions not compatible at the same time. Okay, so it's very much like your house. Your house has 3 environments in it in terms of temperature. The part of the house where you live, that would comprise the bulk of the internal volume of your home. You keep that somewhere between 20 and 22 degrees Celsius. Your refrigerator, which you keep at about 3 degrees Celsius. Okay? We do that because we put food in there so the food won't spoil. Okay? It's short-term storage. Okay? We keep stuff in there. It's not going to last forever in the fridge, okay? But it'll keep it cold. Okay, keep it from spoiling for longer than if I left it out on the counter in the environment in which I live at 21 degrees Celsius. The third environment is your freezer. Okay, that's your long-term storage. You put something in there and it's frozen. Okay, you're keeping that at about minus 17. Okay, most freezers are somewhere between minus 15 and minus 19 degrees Celsius. Okay, if I didn't have those 3 separate environments, I would have to do all 3 of those jobs, which are not compatible with each other, in one environment. Okay, so imagine this. A eukaryotic cell is like your house. It's got the fridge, it's got the freezer, it's got the living area. A prokaryotic cell that doesn't have these compartments has to do all of them in the same place. So it would be your house kept at 8 degrees Celsius. Okay? You'll survive and probably not die of hypothermia at 8 degrees Celsius. Your food will spoil, but not as fast as it would at 22 degrees Celsius, and you basically have no ability to store food long term at that temperature. But it's a happy medium, or an unhappy medium, between the 3 environments. If you're a prokaryotic cell, that's your reality. Okay? You're trying to do 3 incompatible jobs in the same environment. Okay? Everybody with me there? Right. So having those separate environments does mean each process happens much more efficiently than it would in a cell without separate environments. Now, there's a catch. Eukaryotic cells tend to be more sensitive to their environment. That is, they have a very narrow range of environmental conditions in which they will function, because they have to maintain all these internal environments. A prokaryotic cell doesn't. So we find prokaryotes in places where you would never see a eukaryotic organism. We have found bacteria growing in the hot springs in Yellowstone National Park. Okay? Highly saline, so very salty, and very hot. No eukaryotic cell would ever survive under those conditions, but there are prokaryotes that can. Okay? Similarly, we found prokaryotes living in sub-freezing conditions. Okay? Like underneath Arctic ice in water that is incredibly cold. Still a liquid, but incredibly cold and dark. Okay? And they can survive there, where no eukaryotic organism would ever thrive. Okay? So there's a trade-off. Okay? The other thing about eukaryotes and prokaryotes, all multicellular organisms are eukaryotes. Okay? Having separate compartments allows for something called specialization. Okay? Being a eukaryote means not all of my cells are the same. My muscle cells are different from my brain cells, are different from the cells that line my stomach, are different from my skin cells, and so on. I've got lots of different kinds of cells. They're specialized to do a certain job, and that means that they have different amounts of different organelles. As a prokaryote, you can't specialize. All your cells are the same. Okay? You're a single-celled organism, and every other single-celled organism like you is identical. Okay? There's no specialization, so there are no multicellular prokaryotic organisms. Okay? Without organelles, you cannot specialize. Okay? All right. So compartmentalization of cells allows cells to become larger, okay, and more complex, and obviously multicellular. Okay? So if we're looking at a size comparison here, okay, this is a 1/2 micrometer. Okay? Think about how small that is. Okay? The euglena is somewhere between 50 and 80 micrometers. And they were hard to see under high power. Okay? This is 1/2 micrometer. So a virus is like 0.25 micrometers. A bacteria, about 2 micrometers. Maybe a really big bacteria might be 10 micrometers. They're very, very small. Okay? And then you got your average animal cell, which doesn't even fit in the picture. Because your average animal cell is going to be somewhere between 80 to 200 micrometers, depending on what kind of cell they are. Okay, so much, much bigger. Okay, but to be bigger, you got to have efficient processes that can supply fuel, okay, and things like that. All right, questions on compartmentalization? You good with that? All right, so DNA kept where? In the nucleus. It says right there, it's underlined. Okay, so in the nucleus is where your DNA is kept. Okay, so the job of the nucleus is to store your DNA. Okay, and then protein synthesis will be controlled from the nucleolus. So protein synthesis is controlled. You just have to change this from the nucleolus. Okay, so that's that. dot in there. Okay, so the nucleus is usually the largest organelle in the cell. It's usually the thing you can see first. Okay. Kind of a big circle generally in the middle. Okay. Now I was telling you how important its membrane is. It actually has a double membrane that makes sure the only thing that gets in and out is genetic material, and that gets in and out through these nuclear pores. Okay, so the nuclear pores actually look like this under an electron microscope. Okay, so your nucleus looks like this. The little bumps all over the surface are the nuclear pores. Okay, and if you zoom in even more, okay, the surface of the nuclear membrane is intertwined with all these little fibers, okay, that cause that double membrane. All right, now this is not something I'm going to test you like in detail on. You don't need to know all the little details of protein synthesis, but you do need to know how generally it works. So here's how generally protein synthesis works. The nucleolus copies DNA, sends that copy to the ribosomes make the protein, okay? 3 steps, that's generally, okay, how protein synthesis occurs, and that is controlled by the nucleolus. Okay, so the nucleolus produces RNA, okay, which is a copy of the cell's DNA that contains instructions on how to synthesize a protein. That RNA is transported outside the nuclear membrane and joins with ribosomes, okay, that are also produced there, okay, and proteins are created. That has to be performed in the cytoplasm because that's where all the raw materials are. Okay, if you look at the diagram is far more detailed than that explanation. This is the level you need to understand protein synthesis at for Science 10. Okay, when you get to Bio 30, they'll need to understand it at this level. Okay, but that's not for a while yet. Okay, it's basically showing all the things that go on, okay, in this process. So the nucleolus, what it does to your DNA, because it's double-stranded, is it pulls it apart. Okay, and it copies one side because it only needs to copy one side. DNA is complementary, right? The base pairs always go together. Okay, so it just pulls it apart, copies one side, then puts it back together. Okay, sends out the copy through the nuclear pores. You can see that going on right here. Okay, that strand of RNA will go to the ribosome, so it's like messenger RNA. Then it goes to here and becomes transcript RNA, and blah blah blah. It's whole big process. Again, you don't need to know it at that level. Okay, just RNA copies it, sends it to the ribosome. Ribosome makes protein. Good. Okay. All right. Questions on that process? Okay, now the endoplasmic reticulum, there's 2 kinds, okay? Smooth and there's rough. Okay, so within the membranes of the smooth ER, okay, lipids are synthesized, so lipids are fats, okay? So they're synthesized there and then they're transported to the cell membrane, okay, for use in building or repairing membranes, okay? Complex carbohydrates are broken down, drugs and toxins are detoxified. Okay, that's the jobs, okay, of the smooth ER. Okay, hormones can also be produced there. That's less of a job of the smooth ER, okay, but it can do it as well. All right, so if you're looking at what that looks like, the smooth ER looks like this. Okay, the rough ER has all the ribosomes all over it. Under an electron microscope, this is what they look like. Okay, so you can see the rough ER is much more organized. It's got all these layers, and you can see the little ribosomes that are covering it. The smooth ER is just very random. It's just all intertwined and snarled. Okay, so you don't really see a lot of the detail there, but there are no ribosomes on the smooth ER. That's the difference. Okay. When we look at this under the microscope, we will see rough ER. You won't see the smooth ER. Okay? What makes the rough ER visible is the ribosomes. Now, we're not going to see the ribosomes because they're too small, okay? But they help us to see the rough ER. And what we'll see is very much like how this diagram up here appears. If you find the nucleus, it'll often look like the nucleus has a beard, okay, on one or two sides, okay? But if it's just on one side, it'll look like the nucleus is a face and the rough ER is a beard, okay? It'll come off of one side or two sides, okay? So then it could be like mutton chops, okay? But it looks like it comes off of the rough ER, or sorry, of the nucleus like a beard, okay? You'll see kind of a fuzzy, darker kind of structure underneath or attached to the nucleus in some way. That's how you can identify it on Wednesday when we're looking for it. Okay, so the biggest job of the smooth ER is all this stuff about lipids and repairing membranes. So lipids can be moved quickly to the needed sites. Poisons and drugs are detoxified and quickly removed from the cell because the smooth ER runs everywhere and has lots of contacts with the inside of the cell membrane. So there's lots of ways to get those things out of the cell or to the membrane as quickly as possible. All right, questions on the ER? All right, on a test, I would only require one of those functions. Okay, so if you were identifying the smooth ER, you could say synthesizes lipids, repairs membranes, breaks down complex carbohydrates. Any one of those, you wouldn't have to list them all. Okay, as long as you had one there, that would be good. All right, lots of the products from the ER are bound for the Golgi apparatus, especially hormones. Okay, hormones go to the Golgi, and the Golgi can package them in those little vesicles and send them to the membrane. Okay, so we often see the Golgi apparatus drawn like this. Okay, it's kind of this bent half circle made of these different layers. Under a microscope, it looks more like this. Okay, you may see the Golgi apparatus in the cells that you look at. Okay, not always, but you may see it as kind of just a darker, kind of layered, or just a smudge, okay, in one part of the cell. Right? Now, here's what we see with the Golgi apparatus. The one side of it receives the material. That would be over here. Okay, so this is the Golgi right here. Okay, so that's all the Golgi right there. Okay, this is the materials that are coming into it. So this is kind of the receiving side. Okay, as they go through the Golgi, vesicles are created. Okay, these little pockets that you see, and they're shipped out of the cis face or the shipping face. Okay, so all these vesicles come out of one side. It's almost like the Golgi blows bubbles. Okay, and these bubbles just come out of the other side. They contain these materials that came in on this side, and they go straight to the membrane, where they merge with the membrane, and they kind of pop out whatever was in them to the space surrounding the cell. Okay. All right. Questions on the Golgi? Okay, so all it's doing is generally packaging material for export from the cell to get it out. Right? Lysosomes, okay? Lysosomes have to have really, really strong membranes, because inside of lysosomes are digestive enzymes that break down cellular material. Because after all, what is your food made out of? If you eat a steak, what are you eating? Protein. You're eating the tissue of another animal, a cow in particular. Okay? Is that going to have cells in it? Yeah. So your lysosome has to have digestive enzymes capable of breaking down cellular material. That's not the kind of thing you want getting out. If it gets out into the rest of the cell, it'll eat itself to death. Okay? If those lysosomes rupture, the digestive enzymes inside will literally digest the cell. Okay? So we don't want that. So the lysosomes have these really important, strong membranes that keep those digestive enzymes inside, where they only work on the food that's in the lysosome. Okay? That food can be, obviously, food that's taken in from outside, but it can also be used to recycle organelles, because eventually organelles fail. They just break down, they get old, they fail, and the cell will recycle them. It'll use them as food. Okay? It'll break them down, okay, and it'll produce new ones. Okay? The cell is capable of producing new organelles, new mitochondria, and things like that. Okay? All right, so that's the process that goes on. So the lysosome, okay, is when the food is being digested. Okay, digestion can be of the external material or autophagy, which means self-eating. obviously we don't want that to happen. In fact, one of the mechanisms within your cell is basically a self-destruct mechanism. Okay? If a cell detects that it's been damaged and it's malfunctioning, it will, if this mechanism is working, destroy itself. Okay? It will rupture all the lysosomes inside on purpose. Okay? And then the cell will digest itself. Okay? A cell usually does this if some part of its functioning has been damaged and it is now starting to replicate rapidly. Okay? What disease is rapid replication of your own cells? Cancer. Okay? Most of the time, there is a mechanism that cells have that will detect if this is present. Okay? It's usually the result of part of the DNA having been damaged. Okay? If the cell starts doing these kind of cancerous behaviors, it will usually cause its own it'll trigger its own self-destruct mechanism. Okay? It'll rupture all the lysosomes and digest itself. It'll take one for the team. Okay? Now, unfortunately, that mechanism is part of the DNA. If that part of the DNA becomes damaged, then the cell becomes cancerous. It starts dividing out of control. Okay? That's what leads to a tumor. Right? That's the weird thing about cancer. It's why it's so deadly. It's your own cells. Your body doesn't recognize them as a problem because they're you. Okay? The problem with a tumor is it grows fast, and all those cells need energy. So if you have a tumor, let's say on your liver, that tumor will start to grow, and it'll start taking all the raw materials and stuff from the healthy parts of the liver, and the healthy parts of the liver will die as the tumor grows. Okay? That's the danger with cancer, is that those cells grow fast, reproduce quickly, and take the nutrients from the healthy cells, causing the healthy cells to die. Okay? So that's why usually when there's cancer discovered, the first thing that we do is we go in and get rid of the tumor. Okay? They cut out the tumor, okay? And then they give you chemotherapy, which kills rapidly dividing cells, okay? Which are cancerous cells. That's why people who are taking chemo generally lose their hair. Okay? There's rapidly dividing cells that produce your hair. So you lose your hair. Okay? You tend to have a lot of problems producing immune antibodies and T cells and things like that, because those have to rapidly divide. So you become immune compromised if you are receiving chemotherapy. Okay, so those are all things that can be the result of that. Okay? So looking at rapidly dividing cells, if a cell detects it's doing that, it generally will try to kill itself, okay, in order to prevent more of that from being produced and producing a tumor. Okay. The mitochondria. Okay? As we said, the important part of the mitochondria is that it carries out cellular respiration. Okay? It burns sugar and releases the energy within it. No.
Sorry, so the lysosome,
Mm-hmm.
the main thing it does is recycle.
Digest food. Main job, digest food.
This medical center is open 4 times. Can I go answer?
Yes, please do. Okay, now the mitochondria's got that double membrane. Okay, so there's an outer membrane and then there's this folded inner membrane. The folds increase the surface area available for cellular respiration. So the more folds it has, the more cellular respiration it can carry out. Okay, but the double membrane creates a gap, okay, called an intermembrane space between the two layers, very much like how a thermos is designed. Okay, a thermos is designed, it's got the interior container where the liquid is kept, then there's a vacuum between that inside container and the outside of the container. Having a vacuum space in between prevents the heat from being transferred. Okay, so it acts as an insulator, okay, and that's important because the mitochondria produce a lot of heat as they're carrying out cellular respiration. It's why you get hot when you exercise. Okay? When you exercise, you burn more energy, these work harder, and you get hot. Same with if you run a fever. Okay? If you run a fever, your body has triggered these to run hotter than normal, to burn more sugars than normal and raise your body temperature. Why does your body do that? A fever is your body's natural reaction to something. When do you get a fever? When you're sick, when you have an infection. Okay? The reason your body does this is because whatever's infecting you likes it in your body the way your body is, 37 degrees Celsius. So, your body says, fine, you like it here? I'm going to make it really unpleasant here. I'm going to make it way too hot for you. Okay? And it'll spike your temperature. Okay? When your temperature spikes, all the reactions that allow your cells to work stop working. Okay? They don't completely stop, but they don't work efficiently. They're designed to work at 37. Same as the bacteria or virus or whatever that's infected you, okay? It works ideally around those temperatures, especially if it's a virus which is just using your own cells to work. Okay? If you raise your temperature, your cells aren't even working all that well anymore. Okay? That's why when you have a fever, you feel just bagged. Okay? Your body is not working well. Okay? All the stuff that your body does normally to keep you going is working at a much lower rate of efficiency. Okay? And that can slow the infection. Okay? Now, obviously, it gets to a point where it can be unsafe for you. If you're running a really high fever for several days, that can be deadly. Okay? So, then we would have to bring your temperature down. But over the short term, it helps your immune system catch up, because it slows the growth of the infection. All right, so mitochondria carries out cellular respiration. That's the big thing for those. Okay? All right, chloroplasts. Only going to find those in plant cells. What do? Right. If you were labeling this on a test, I would see chloroplast, and then in the function box, photosynthesis. That's all you have to write. That's all they do. Okay? They carry out photosynthesis. Okay? They also have a double membrane. Photosynthesis can produce some heat as well, so they have this double membrane that kind of insulates them a little bit. All right, and then we got these other organelles that we'll just kind of quickly talk about here. Okay, first one, cell membrane. Remember I told you it's like Jell-O salad, it's not like saran wrap. Okay, this is what your cell membrane really looks like. Okay, so you can see the double layer of fats, all the little white balls, those are fat molecules. They're turned in so their tails face each other. That creates that water tight seal essentially against the outside of the cell, right? But embedded in that lipid bilayer, okay, or double layer of lipids, are all kinds of things, okay? Receptor proteins, glycoproteins, all kinds of stuff is embedded in there, okay? It looks literally like Jell-O salad. It's a gelatin, okay, that's got all kinds of stuff in it, okay? And all of these parts perform a different job, okay? They can be receptors for hormones. They can be receptors that trigger the absorption of food and water, okay, or other things like that. So there's all kinds of things. The membrane is actually a very complex place. Okay, so cell membrane, and these are the two important words. If you are telling me what the cell membrane does, I will need to see those two words. Okay, the cell membrane controls endocytosis and exocytosis. That is movement in and out of the cell. Okay. What I don't want to see is protects the cell, because it actually doesn't do that. There's lots of stuff that can get through the cell membrane that can hurt the cell. Okay? It controls what gets in and out. That's it. Okay? Endocytosis and exocytosis. Make sure you use those terms. Those are the scientific terms. Okay. The cell wall. Right? We said before, it's not really a wall, it's more of a frame. Okay? This is what the cell wall looks like under an electron microscope. Okay? This green part, that's the cell wall. Okay? It's hollow, full of holes, both ways. Okay? It's just a frame. It's there to hold the cell's shape, not to act as a border guard, not to do any filtering, not to do any protection. It's just a frame. It holds the cell's shape. Okay? And that's all. And it's made out of cellulose, which is that fiber we were talking about, okay, that we can't digest. Okay? Herbivores can, but we right? And then we've got the water vacuole, okay, which is just the big bubble of water, okay, that's inside plant cells that works with the cell wall using turgor pressure, okay, that's an important term, okay, turgor pressure, okay, of water, okay, supporting the cell from within, pushing everything against the frame, okay, makes it stronger. Okay, and then we got the centrioles, which we said were only in animal cells, and they help with the cell division. Okay, this is what they look like. Okay, so they look kind of like these little gears, and I was telling you that during cell division, they would migrate to the poles, and then they'll put out these little fibers that'll grab onto the chromosomes and pull them apart, okay, making sure that each new cell gets all of the genetic material that it needs. Okay, so that's what they do. So most of the time, your centrioles are inactive. They just sit there and they don't do anything until it's time for the cell to divide. All right, and then lastly, the microfibers, the microfilaments, we summed that up into one term, the cytoskeleton. Okay, and that helps the cell stay intact during changes in shape, also only present in animal cells. Okay, questions on any of that? All right, tomorrow your microscopy labs are due. Hopefully you're pretty close to done those. I will be posting your Tuesday quiz this afternoon. Guess what will be on it? Parts and functions of the cell. So make sure you have a look over the diagram, okay, tonight, so that you know what to do. All right, if I can get you guys all to pack up, and then I'll come around and spray your desks.